Thank you. I'm humbly honored to be talking here at the Santa Clara Native Plant Society. I had an opportunity when I was a graduate student. Before I went to UCSB, I got a master's at Oregon State. And I had the chance to be the field trip coordinator for the NPSO, the Native Plant Society of Oregon, the Corvallis chapter. And it was such a relief to step out of the laboratory, the greenhouse of my academic ivory tower, and be with the community, in the field, amidst people who were truly passionate about plants. And so I have the utmost respect for you, and hearing the announcements here and all the things that are going on and reading it in the Blazing Star is really motivates me to try to share that with students at Santa Clara. So I thank you guys, and like I said, I'm humbly honored here. I brought some show and tell. These ones are not for sale. They're part of an experiment, in fact. I snuck them out underneath my undergraduate's eyes, so make sure you don't take them home. As we go, I will try to explain, but you're welcome to come up afterwards. I brought some soil samples, some columbines, and even a couple pollinators. What I thought I would start with is something that makes Santa Clara Valley unique. The Silicon Valley for some, the Blossom Valley for the rest of us. And in fact, before it was the Blossom Valley, it was a different kind of floral valley, right? In fact, it's one of the most distinctive places in temperate North America, particularly because of the steep ecological gradients. That just means you can go from one habitat into the next in a very short distance, and that makes the Santa Clara general area extremely unique. Take, for example, you can be in the wettest forest here, the Redwood Forest of North America. Technology fails, but botany rules.
Can we get the lights down more?
Yeah.
Mm-hmm.
Like, for example, here at Henry Cowell Redwood State Park, you can see one of the wettest forests in western North America, yet immediately adjacent to some of those forests, you can find some of the driest temperate non-desert areas like Chaparral. Take, for example, that familiar Mount Omanum, which we'll soon have access to, I understand. Yet this is an axis of moisture. And I'd like to expand your minds into another axis of ecological gradients, and that is the chemical axis. Take, for example, what's going on in the soil here at Henry Cove. You can see a serpentine outcrop, and something unique about those soils is preventing a large number of those plants from occupying them. Yet at the same time, those areas harbor amazing amounts of diversity, which I'm sure many of you are familiar with. Yet not all serpentine habitats look like that. That's probably the most famous, as you've seen from roadsides all over the county, but occasionally we will have serpentine seeps in Santa Clara County, and they are more cryptic. The chemical axis is hidden in the soil, but this is Santa Teresa Park. It's the Stiles Ranch entrance, and there's a giant serpentine seep here. Maybe some of you have been to Santa Teresa Park? Yeah, excellent. Okay, so you all are familiar with that area. In fact, the diversity that is harbored in those serpentine soils is amazing. So there's about 1,400 endemics to California. That means plants that only grow in California. And of those, approximately 180 of them are serpentine endemics, can only grow on serpentine soil. If you do the math, 180 of those, I said, right, is 13% roughly. Yet, they occupy only 1.5% of California's soil area. Okay? So we have packed into 1.5% of California some 13% of endemics. This is amazingly interesting for me from a diversity angle, and we should be very proud of that, and in fact be attempting to conserve. Yet there must be something really unique going on, and how have these plants gotten here? Well, I figured if I put up something about chemistry in the periodic table, I'd better do it in the first couple minutes. And save the pollinators in the movies for the last few minutes. So hopefully this doesn't bring back too many nightmares. But obviously we're talking about the chemical axis of the soil, and there's a couple very unique things about serpentine that you probably have heard of before, which is there's something unusual about the levels of calcium and magnesium. Now, calcium is essential for plants to grow. In fact, it's involved in the cell wall expansion. Yet, plants are blind to what element they're picking up. They only see the +2 charge, which is this entire column here. Serpentine soils, of course, have high magnesium and low calcium. Blind to that, most plants' roots will absorb magnesium at 5 to 10 times the concentration of calcium. meaning that they're taking up 10 atoms of magnesium for every 1 atom of calcium. Not only are they not getting enough calcium, but that magnesium is toxic to most plant roots. Not all plant roots, but most plant roots. In addition to the calcium-magnesium ratio that makes serpentine soils unique, there's also some heavy metals, nickel, for example. In particular, around Coyote Ridge, we see nickel levels that are 10 to 20 times higher than you see in non-serpentine soils in the adjacent areas. I'll show you some of that data later. We also see low levels of nitrogen, phosphorus, potassium, and very low water holding capacity in some serpentine soils, but clearly that's not true of a serpentine seep, which will have wet, mucky soil in the middle of August, right there at Stiles Ranch if you've been by that little switchback area. So knowing a little bit about the chemistry, it's not surprising how challenging it is. Yet, a large proportion, 180 plants in California, have figured it out. And they're not all dicots. They're not all flowering plants. In fact, we see endemics like this. You recognize this, anyone? It's a cypress, and the Serpentine endemic is Sargent cypress, or this one is Sargent cypress. And down here is a Serpentine endemic. It's a fern. Anyone? It's related to the sword fern, Ballisticum. This is Ballisticum lemonii from the northwest area. We also see the Metcalf Jewelflower in the top left there, one of the topics of today's talk, and the Serpentine Columbine. But my main point here is that Serpentine endemics actually span the range of land plants from ferns, gymnosperms, and monocots and dicots. And if you're fascinated by this type of diversity and you want some new references, there's been some great work that's come out, and I don't want to go into detail, but I wanted to point it out in case you have access to this. In a recent paper, 2005, by Hugh Stafford, and out of Susan Harris's lab at UC Davis in Madronio, he has done a revision of Art Krukeberg's famous work on serpentine endemism in California. You're probably familiar with Art Krukeberg, and as far as I'm concerned, Hugh Stafford is following closely in his footsteps. He actually realized that serpentine endemism is a range. And in fact, he scored them based on how confident we can be that they are truly only on serpentine, or maybe sometimes they are tolerators. They're found on and off of serpentine. As anyone who's looked at these plants realizes, sometimes there are. And I just grabbed the top couple families from this list, as it's fascinating to see that although the sunflowers, the lilies, the mustards, scroffs, and carrots are high on the list of serpentine endemics, when you compare them to California endemics, so California endemics in brown bars, serpentine endemics in white bars, you'll see that often the serpentine endemics are disproportionately represented. For example, in the lilies and the mustards. In the mustards is particularly the jewelflower genus Streptanthus that is driving that. I encourage you to look further at that if you are interested. I'm not going to talk in any more detail, except that a friend of mine, graduate student, Brian Anneker from Susan Harrison's lab at UC Davis, we put these data points to work. We found some evolutionary relationships amongst 25 California genera. You guys maybe know Navaricia. In fact, there's a couple serpentine endemics in Navaricia. And we were asking some big evolutionary pictures about the evolutionary history of serpentine endemism. The take-home lesson here is that the serpentine endemics, they're pictured here in the darker gray, tend to evolve out of serpentine tolerators, not surprisingly. Something that can grow on and off serpentine tends to bud off a species that's an endemic, and both of those tend to come from non-serpentine ancestors. But what's fascinating for us, and maybe for conservation, is that these lineages tend to not speciate as fast. In fact, they're more sensitive to extinction. Not human-induced extinction, but in particular, we're picking up evolutionary time points measured on the order of 1 to 10 million years. So these guys are already naturally susceptible to extinction, as you might expect these little serpentine island-like habitats would lead to. We didn't just find that in Navaricia, but we added Collinsia, Erythronium, Allium, and we find repeatedly the lack of the ability to evolve out of serpentine. So these are very fragile habitats to start with. And then we compound it with development, and it becomes, for example, nitrification, and it becomes extremely high conservation priority. I encourage you to have a look at that if you want more details. It's in Evolution in 2011. But what I'd like to tell you more today is less mumbo-jumbo about phylogenetic trees and more stories about plants. In fact, I have 2 stories in particular that I want to focus on. And I realized that, in fact, Judy and Arvin were very diligent about checking and rechecking the poster, the advertisement here, that said I will talk about 3 local Serpentine endemics. In fact, one of them I wanted to talk about was the San Benito evening primrose, Camusonia benetensis, from the BLM area near Hollister, yet of course not in Santa Clara. Therefore, the local part was not really true.
We can check field trips there.
Ah, excellent. Well, it's too late because I chucked it. I'd be glad to talk in more detail about it. In fact, the work is ongoing in the lab, and we're finding some really interesting things about that plant. But the Columbines and the Jewelflowers are really exciting, and I wanted to make sure I had enough time to tell their story. So for that reason, again, come talk to me afterwards if you want to know more details about the San Benito evening primrose. That's a project that's still ongoing in my lab. So here's where I'd like to go today. First, I want to ask the question and hopefully provide some details on whether or not the Metcalf Jewelflower is evolutionarily distinct. Is it a real lineage? Is it a species or a subspecies? Does it deserve protection? And can we detect this using evolutionary and ecological experiments? And second of all, I want to test what is the role of adaptation during speciation in the serpentine columbine, that's Aquilegia eximia. In particular, this is a contentious issue in the field of speciation, but it's also really fun to do. You get to grow the plants on serpentine and non-serpentine. You get to go out and look for pollinators and see where the pollen lands on these hummingbirds. And so I took on these projects because they're local, and I hope to be here for some time in the future. And I hope to continue the studies on these guys. So let's start with the Metcalf Jewelflower. If you're not familiar with jewelflowers, you'll realize these are exceptional mustards in that the majority of the show are the sepals. They're kind of balloon-like, they're expanded, and they often come in different colors, and the Metcalf Jewelflower is distinctive in having almost purely white to cream, sometimes slightly yellow or greenish sepals. That's very unusual for mustards, where usually the majority of the show is in the petals. In fact, if you stare at them long enough, you'll see there's kind of a bilateral symmetry. They've taken on sort of a two-lipped, more of a mint family or figwort family shape, rather than the typical cross shape of brassica or radish, for example. Is also a serpentine endemic, restricted to the central northern portions of Coyote Ridge. So you're all familiar with this portion of our area. It's such a sensitive place and an exciting place to do research. In fact, the central and northern portion of Coyote Ridge will have only Metcalf Jewelflower. Yet you'll start to see pink and purplish individuals around Kirby Canyon and the Butterfly Preserve as you move southward there, and the UTC land that I guess has recently changed hands for the better. And then as you go further south and the surrounding area, you'll see the flowers turn purple, and we call that Streptanthus glandulosus. I'm going to gloss over the taxonomy. There are some controversial new treatments in the Jepson manual that nobody checked with me, but only because I sense that the new names are not substantiated by science. One of the important things when we're voting, yes, from our earlier announcement. In fact, science should be used in determining the names, and I would just like to present some science today and then let you guys decide what the appropriate name is. As you are not tied to the Jepson manual treatment as long as you cite the author that you are tied to. And the Jepson Manual #1 did a fine job, I felt. Without any more controversial statements, let's get to the science. You may recognize this road cut on Metcalf Canyon. In fact, it's on the way up the road from 101 to the motorcycle park and eventually to the dump. It turns out that there are a couple of jewelflowers along here. They love these roadside cuts. Extremely disturbed, south-facing. You couldn't get more miserable habitat. Plus, the calcium-magnesium ratios, the nickel ratios, etc. I just want to show you where some of this work is done. It's done in this small divide between the safety zone and the dump truck. So there are a lot of multiple uses going on in Metcalf Canyon. Here's Metcalf Road. You'll see the diversity of multiple uses of that area. In fact, some of them are not so official. Here's the Metcalf Jewelflower, doing quite well. And down here you'll see a bed and a refrigerator. And a lot of people just can't make it to the dump, and unfortunately. I'll also point out the power lines here, as this is the western extent of the motorcycle park, which seems like potentially conflicting use of the land, although we've had an excellent stewardship up there, and I'd love to talk with you guys later about that. But they have a great little rangers program. They tend to stay on the trails, and the jewel flowers do quite well there. It could be a lot worse. On to the science. Here are the old treatments of the 3 jewel flowers that we're going to focus on. This is the Metcalf Jewelflower, Streptanthus albidus, subspecies albidus, for its white sepals. And subspecies Paramoenus comes in a range of pink and purples, and Streptanthus glandulosus tends to have a darker purple, slightly obscured by the image there. But we know that these flower colors are not qualitative, but in fact they can be quantified. In fact, we should probably be careful to think of these flowers in terms of what the pollinators see. We think the primary pollinators are probably insects, so we can go into the UV. We can do that using a portable spectrophotometer. And over here we're looking at how much light is reflected in percentage, and down here we're looking at the wavelengths of light. And just to orient you, humans can see from about 700 to 400 with our trichromatic color vision, and bees see a little bit deeper into the UV, yet lack some of the ability to see the reds. The main point here is that there's not very much reflectance in the UV, but as soon as you get to the visible, you'll see the reflectance comes up, and white has this kind of characteristic slow hum to it. When we add some pigment to it, it turns out those pigments, they capture light and they hold it. They pull it down so that it's not reflected. And when we compare it to subspecies Paramoenus, you'll see that the curve goes up a little higher. And these are the pigments, the anthocyanins, that are pulling that light down. The main point here is we have an average line, that's in bold, and a standard error line around that. And this has been sampled over 20 times for each plant, for each species. And so you see there's a real significant quantitative difference. That's my emphasis here. If you add in Streptanthus glandulosus, you'll see it's a much darker flower, pulling a lot more light down across all wavelengths, but particularly here between the 500 and 550s. Neither of these have any dramatic reflectance in the UV, so it doesn't seem like there's any spectacular show that we're missing. Now, atop Mount Hamilton, around the observatory there in the shale, there's another jewel flower that was somewhat mysterious, so we added it to the study. And sometimes it goes by the name of Streptanthus glandulosus, and it's been given other names. And it has a very, very dark, almost brownish, merlot-colored sepals. And you'll see that as you get further and further into the dark, dark colors, less light is reflected. So there is definitely a significant difference in the flower colors in a quantitative fashion. That seems like justification alone for calling these things unique. But some people argue that sometimes these flower colors can change because of a single gene. So what? It could still be a new species. We know that single genes cause differences between other species, like, say, the monkey flowers from the Sierra, Mimulus lewisii and Mimulus cardinalis, which we have around here. Regardless, let's look at some other traits and see if these plants are distinguishable. Here's one of our very own, Don Mayle, and there's Stu Weiss, the famous Stu Weiss, and Sharon Strauss from UC Davis, and we're up atop Coyote Ridge there collecting seed and soil for a comparative study of the soil chemistry, wondering maybe these plants occupy unique soil types, even though they're all serpentine. Here's the calcium-magnesium ratios, and we have the averages. You'll see they're all below 1. Meaning, on average, there's more magnesium than there is calcium. In fact, some like 5 more molecules of magnesium to every molecule of calcium. And the error bars represent multiple samples, and you'll see there's overlapping error bars, and there is no significant difference between any of the 3 samples, nor Tulare Hill, which had once Metcalf Jewelflower and no longer does, and you can barely not quite see it off the edge of the picture here. This is looking northward, northward on 101 and Santa Teresa in particular. That Tulare Hill is going to come up repeatedly as a potential reintroduction site for this plant. The main point here is that there's no dramatic difference in calcium-magnesium, yet maybe some other more cryptic chemistry differs between them, so we did a battery of chemical tests on the soil, some 25 different chemical and physical characteristics, and we were able to come up with this principal components analysis. It's a little bit of a complicated statistic, but the main point is that it's taking all of the data and trying to spread it out into just 2 dimensions, because we have like 25 variables, and it's hard to visualize them all at the same time. So in 2 dimensions, we explain some 50% of the variation. And here's the albidus samples in the white triangles, in the pink paramoenas, and you'll see substantial overlap in the chemistry. And when you throw in the glandulosus, they're also significantly overlapping, not significantly different. And what was most fascinating was that the reintroduction site had very, very similar soil too, such that it doesn't look like there's dramatic differences in the chemistry in trying to find things that make these species unique. Yet, to assume that we could measure every possible chemical that a plant can taste is an overstatement. So why don't we ask the plants whether they can tell the difference? These are the means, if you want to put the means on here, but the error bar's all over them. So we went back out. That's Sharon and an undergraduate at the time, Justen, who now just got his master's degree from Alaska. And we went back out and collected some seeds as well as soil, and we were going to do a transplant study and ask if the plants prefer their home soil, where they came from, or whether they do better in another species' soil. And if we see a lot of home soil advantage, that would suggest they're adapted to their own soil, and there's some ecological uniqueness there. If they are not, then that would suggest otherwise. So we grew a lot of jewel flowers at the UC Davis Lath-house. This has slats here. Each one of these jewel flowers is sitting in a container. The container racks are about 2 feet by 3 feet, and you'll see they're in these tubs. The great thing about growing jewel flowers is that you only have to water them once a week, and sometimes that's too much. That's in the Lath-house in UC Davis in the middle of July, which if you've been to Central Valley in July, you know what that means. So these are extremely drought resistant, and we were able to get them up and growing and flowering within just a couple of months. And we counted the dry biomass, here measured in grams. And I'm first going to show you the result when we planted the jewelflower, Metcalf Jewelflower seeds, onto their own soil, onto the paramoenas soil from Kirby Canyon. Then we planted it onto glandulosus soil from the Santa Teresa area. And you'll see that, in fact, they don't prefer their home soil. Remember, that this is that horrible south-facing exposed road cut. Actually, we went out to the motorcycle park, but it's just as miserable. And as you get to the reintroduction site, you'll see actually the plants grow larger. And in fact, they also make more flowers. The data looks very similar to this when you count the number of flowers that they produce. So it looks like no evidence for a home site advantage here. In fact, what was really surprising was when we compared what happens when you plant the paramoenas seeds, those are the pink ones from Kirby, the graph changes subtly at best, right? Similar pattern. That would be, even paramoenas doesn't prefer its own soil, it would prefer the glandulosus or the Tulare Hill soil. Now that Tulare Hill soil has had cattle on it at differing rates of, differing densities of cattle, and that could be contributing, as we do see slightly higher nitrogen out at Tulare. It's still a potential reintroduction site for us now that we've got the cattle grazing regimen in hand, and we have yet to find a density of some of the particularly bad invasives. Let's move on to reproduction. Maybe the plants are different in how they reproduce, because it's definitely not the soil type that causes it. And so we spent quite a bit of time looking for pollinators, and in fact, before we went to the pollinators, we were looking at how many fruits they produce. It was really astounding at how many fruits are produced on a single plant. You'll see here's the inflorescence axis. Here are the open flowers. And each one of these fruits is developing, which was really astounding. They can get fruits to about this long, holding between 20 and 30 seeds each. It was particularly astounding because in the Lath-house experiment I showed, we grew over 1,000 plants and found only 12 plants would produce a seed. Now, for conservation purposes, and particularly the idea of bulking seed for reintroduction, this is a big problem. Now, the Lath-house has slats, but pollinators could go in and out. So the thought was, well, you know, there just weren't enough pollinators. We also did the same experiment in a greenhouse and found less than 1% fruit production, and that's indicated here. In the wild, compared to the greenhouse, the proportion of flowers with matured fruits, and you'll see they produce, you know, 75%, 70 to 75% fruits in the wild, yet they barely ever make a fruit in the greenhouse. And the greenhouse plants are quite happy. The flower colors are consistent in the greenhouse, yet there are no pollinators, suggesting that these plants rely heavily on pollination services. And if we're going to conserve the pollinators, then we're also, if we're going to conserve the plants, then we've got to conserve the pollinators if we want these plants to make seeds. It looks like the most common pollinator is Bombus vosnesenskii. This is its better end, as this is the end you use to identify it. And in fact, you'll see we use digital camcorders to do our pollinator observations, as I have access to a lot of very passionate undergraduates who bring the same sort of motivation as you guys do, except they're sometimes a little less familiar with the plants, let alone the pollinators. Although I know we have one in the audience who knows a lot about pollinators. Anyways, using digital camcorders, we can set these up. The students can run around Coyote Ridge, changing the battery, changing the tapes. At the time, we were using little mini DV tapes. We now switched over to hard drives, but regardless. And then we bring it all back. They pop some popcorn and sit and watch hours and hours of pollinator movies. It sounds really great in the beginning, doesn't it? Till the popcorn runs out and you still have 98 hours of pollinator movies left to watch. Regardless, they were extremely helpful in analyzing this data. And here is our three of them, Stephanie, Peter, and Manny, posed here over their digital camcorder with some Metcalf Jewelflower. We're able to watch 5 or 6 flowers at a time, sometimes an entire inflorescence. And the great thing about this, especially using the camcorders and the undergraduates in the field, is that we can set up multiple camcorders on these outcrops. Here we're at the motorcycle park, and if you squint, you might even see the tripods. We had 5 camcorders going from sunup to sundown, and we can collect hundreds of flower hours in just a couple of days. A typical study would study 10 or 15 flower hours, and that'd be a pretty thorough examination. And meanwhile, you know, I get to sit there and eat chips and salsa and manage the whole thing. It's wonderful, especially after they get the movies, then they can come to me. But in fact, we're really busy at that time because we're trying to make sure we have documented pollinators. We do have to catch a couple of them and identify them appropriately. So that part's just a trick. Here are a couple of stills, and if everything's going correctly, we might even get a chance to see. That doesn't seem good. We captured these on mini DV tapes, and we now have a whole handful of mini DV tapes. And on the fly here, let's see if we can get this movie running to show you the most common pollinator. Here, taken from the frontage road right in front of the UTC property. And that's Bombus vosnesenskii, and there goes the vehicle. And you'll see it's moving within a flower, within a plant, amongst multiple flowers. Bumblebees typically move from the bottom of the inflorescence up. It might not be surprising that most flowers are male at the male first and then female later, such that pollen would be moved from the last plant onto the stigmas of a new plant.
Squashes like that.
Yep, a lot of plants will be, so the female flowers will be open at this phase here, and there'll be male at the top, and then theoretically those pollen goes on to the next plant, and they land on a female. And that encourages outcrossing and theoretically preserves some genetic diversity. We watched, like I said, over 100 flower hours of this plant, and we found 99% Bombus vosnesenskii. It's unbelievable how common they are. I think if you look carefully, you can even see a couple in the background in earlier frames. A couple honeybees, there's another one. A couple honeybees came in, and we have occasional, early in the year and late in the year, we'll see circuit flies and some solitary bees. But the majority of the brunt load that's producing the 70 to 75% fruit set is this service by these bumblebees, and it's unbelievable how many there are out there. So we went on to ask another question, because it turns out Bombus vosnesenskii is not just the primary pollinator of the Metcalf Jewelflower, but also the movie you're watching was actually on the subspecies Paramoenus. It was a slightly pink-tinged flower, and that's right along the road there. And it turns out that Bombus vosnesenskii is also the primary pollinator of that, and Streptanthus glandulosus. So it doesn't look like there's a major pollinator shift driving that. Like in my Columbine work, we found shifts from bee to hummingbird and hummingbird to hawk moth. Of course, these subtle mustard colored shifts are not going to cause such dramatic pollinator preferences. So the last attempt here was to try to find differences between the species using molecular markers, using DNA, because DNA contains the footprint of their evolutionary history. And it's a little complicated, but I just want to point out the take-home lesson here. You can imagine this is like the tree of life for jewel flowers. Okay? And it grows from the bottom, this is the root, to the top, and these are the tips. And each one of these names is a plant, which we took a leaf off, and students and I extracted DNA from it quite readily, and were able to examine some 600 data points across all of its chromosomes. So we're not just looking at one little piece, like the flower color is probably just one gene, but we look throughout its chromosomes at about 600 pieces. And of course you can't make out any of those names, so it means nothing. But these are all the Streptanthus albidus, subspecies albidus, Metcalf jewelflowers. And it turns out that they all come from a single common ancestor, which suggests they are a unique evolutionary lineage. Like all humans come from a single common ancestor. The Paramoinus, although they have a common ancestor, they seem to have budded off this species, the Metcalf jewelflower. Okay? This is the Kirby Canyon area, for example. And the common ancestor of all of them is Streptanthus glandulosus, which is also a very widespread species. Now, the data...
Excuse me,
Yep.
Justin. So the leaves at the top of the graph are essentially individual plants.
Exactly.
Okay.
Exactly. Individual plants from Motorcycle Park, from Stiles Ranch, from Kirby Canyon over here. Okay? And the main point here is that they do cluster together. Suggests they're evolutionarily distinct. But it's subtle, I have to be honest. This wouldn't be particularly dramatic results, as the support values are weak. That's these numbers here. And we only trust the numbers bigger than 75 to 80, and very few of these branches showing no numbers had values higher than that. What did pop out from this, fascinating, is that the plants from the top of Mount Hamilton, from Observatory Peak and Tortilla Flats, Tortilla Flats is a little side road there where some of the residents live, are very distinctive. They're on their own evolutionary lineage over here. They also have a unique flower color. It turns out the soil up there is not serpentine soil. It has a very different chemistry. So I leave you here with the still question of whether or not the Metcalf jewelflower is an evolutionary distinct thing. If it is, because of the flower color and some of this DNA evidence, then it's an extremely rare plant. It's only limited to the central and northern half of Coyote Ridge. And these habitats are being destroyed. For example, one of the main habitats is the Silver Creek Golf Course, and you have to sort of fend off golf balls in sampling those plants. Stu Weiss and his group and I have put together a proposal to attempt to build an entire new population. This is on the margins of my expertise, but it's very exciting because the proposal is to actually go out and collect a few seeds, use those in the greenhouse to generate plants, some 200 plants, which we estimate could produce tens of thousands of seeds, only if we take them back out there so the pollinators can do their work, right? Which is much better than me having to make all the individual pollinations. Once the plants make their seeds, we'll use those seeds in the reintroduction in a very controlled way to figure out which habitat parameters that they like. And then we estimate we could generate a population of about 8,000 to 10,000 with very conservative survival. That's 1 out of every 100 plants planted survived. We think we can produce a very large population. And if we can get that size population, it would have the potential of producing millions of seeds. Within 4 years is the attempted proposal.
Is Bombus out there?
Yes. So Motorcycle Park is right here, and all of our pollinator work was done right here. And we've seen Bombus vosnesenskii out here, and it's a very short drive, assuming you don't get hit by a car along the way. Tulare Hill is bordered by Santa Teresa, and that's Monterey Road right there, yeah? And there's the power plant, and it's in conservation hands. So we plan to use the sites on the south end of Tulare Hill, facing north here, as these reintroduction sites. And then we need a control site to make sure that if they fail here, we know why. And if we plant them back out where they actually exist, we could test for whether or not it was our planting or whether it's the site that they don't like.
Let's hope you have a decent year, not like what
Keep in mind, this is a mustard. You've surely grown some mustards. Mustards do the exact opposite as all of these. When it's a bad year, mustards do wonderful, particularly these mustards on south-facing slopes with exposures like this. I'm talking particularly about the Metcalf jewelflower. When it's a great year, when it's really wet, for example, they actually have a hard time. I think it's predators, for example, pyarid butterflies. These cabbage butterflies will go through a big year when there's a lot of moisture, and the caterpillars will consume a large number of plants, at least on Metcalf Road. So this year might be a great year to drive out to Metcalf Road and see if you can find some jewel flowers. So hopefully this sort of brings it round circle from some sort of semi-esoteric experiments, some ecological and evolutionary experiments, to actually, muddy boots biology, actually getting out there and starting and establishing new populations.
Question.
Yes?
Is this the west side of the ridge?
Tulare?
Yes.
Here, this is the south facing. So 101's over here, and San Jose is right on the other side.
That'd be like from Bailey.
Yeah, the picture was taken from Bailey. Thank you.
Question?
Yep.
So, did I understand it right that your plants in the Lath-house didn't produce seed
Correct.
because this bumblebee was not there?
Yes. That's what we infer. We haven't done...
So there are no other pollinators for this plant besides the bumblebee?
That's not necessarily true. We just know in the wild, this bumblebee is extremely common. And in the Lath-house in Davis, it was not common. In fact, very few bumblebees make it into the Lath-house because it's not a well-known place for nectar. It's in the middle of UC Davis. So what we'd have to do for this reintroduction is to grow the plants at Santa Clara University, for example, until they are ready to flower. Then we would take them all out to the field. We'd bury the pots about halfway in so they don't get tipped over, and they would flower, and the bumblebees would do their magic. In fact, we would do that in an existing population. We collect all the fruits of that, and that way we have a minimum impact on the wild population, and at the same time we generate a huge number of seeds for the first years of introduction.
So in your test, the inside test that Davis, were you hand pollinating?
No.
There was no attempt to...
That's the next experiment to do, would be the hand pollinations to prove that outcrossing...
My guess would be without pollinators, you wouldn't get very many seeds.
Exactly.
So since the bumblebees are pollinating all three species, do you have evidence that the three species intercross?
Yes.
fairly easily?
They're definitely infertile in the greenhouse, okay? But whether they cross in the wild is a big question. And it's a conundrum for me. If you ever drive out there, you'll see the pink ones are stuck on Kirby Canyon, and the white ones are stuck at Metcalf Road. And in between, there is a small zone, or on the UTC, the old UTC property, where you would find some white ones and some pink ones mixed. But it's a very, very quick divide between the whites to the north and the pinks to the south. And the cause of that, in my mind, remains uncertain. Yeah?
What are the species of butterflies that are eating the caterpillars?
Yeah, we see mostly pyarid, little cabbage moth. Yeah.
Just plain cabbage or some of the native pairs, do you know?
I'm not the one to ask for that. I have to be honest with you. I have some photos that if you know the difference between them, maybe I could send them to you.
Yeah, I'd like you to do that.
Okay.
Thank you.
You can identify them from caterpillars.
Okay, you have the caterpillar
Yeah.
photo?
Yeah, Don.
Help me here. I have seen on UTC amongst the pink every once in a while a white.
Yep.
And one on the access road at Kirby Canyon, one white and nothing more than pink. Any
Right.
idea what's going on there?
Yeah, there's no doubt that over both historical time periods and over evolutionary time periods, seeds, these don't have spectacular wings. They do have a small marginal wing on the seed. The fruit doesn't have any major animal dispersal mode, but it would eventually get stuck in the fur of some of the larger grazing animals there and move around. It's over millions of years, for example, seeds should move back and forth
So
between
we
the
got one
combination.
seed. We got one seed on the access road,
Yep.
and maybe five.
Maybe it was on my truck. That's my biggest concern.
Before you got it.
Good strong wind.
Yep. In fact, there was the first Metcalf Jewelflower in some 30 or 40 years was found at Tulare Hill, and the cattle that are grazed there come directly from there. And I'm sure that those cowboys are not washing off their lips in transport. And so there's no doubt that those plants can move around. But the question is, why is it not a larger mixed population? And from an evolutionary perspective, that remains a large conundrum and something that's fascinating for me. And I was hoping there would be some correlation. White flowers do better on this soil, and purple flowers do better on this soil. But as a scientist, I have to acknowledge that's not the case. Yeah, no.
So did you do any What kind of results did you get from the soil samples on the Mount Hamilton jewelflower? Was it still in that 2-electron?
It's way off the scale here. So, right here. So here with the calcium-magnesium ratio, and take a look at the y-axis, okay? And so it doesn't even get up to 1. And most normal soils are more like 4 or 5 molecules of calcium for every 1 of magnesium. So these blind plants would get 5 calciums or so for every magnesium. When you throw in the Mount Hamilton soil, although it's very low in nitrogen, it's basically a shale up there, the axis has changed, and this is the Mount Hamilton plants. What they are remains uncertain, but there's no doubt that they are very different chemically in that particular way. There's also no nickel in those soils, or no detectable nickel at the measurement of parts per million. But they are a challenging soil for water holding capacity and for some of the basic nutrients like nitrogen. But calcium, no problem. A couple of things that we bumped into that remain unknown are what's going on here at the top of Mount Hamilton. Thank you very much. In fact, right here by the observatory, right on the road and around the parking lot, are these jewel flowers, and they have these unusual sepal colors. They're particularly dark. They have some distinctive soil chemistry like we just saw. In fact, they're even growing out of old brick walls. You can see the infructescence. This is the fruiting stem, and one of the observatories are right there. It's definitely ecologically differentiated, as we could not get them to grow. In the reciprocal transplant in the Lath-house, they would not grow. They continued to die, and we kept replanting them in the serpentine soil. They are not adapted to serpentine, and on the DNA level, they are on a unique evolutionary branch. So if anything should be recognized as a new species, not so much the Metcalf Jewelflower, but definitely more investigation needs to be going on with these dark colored ones. In fact, on the small canyon that leads to Grant Ranch, I saw on the road there more of this dark sepal jewelflower, suggesting that it's not just restricted to the very top here. And in fact, just before this week when I was preparing for this, I typed in Mount Hamilton jewelflower, and I got something from Hollister that looks suspiciously similar with these chocolate-colored sepals. So it may be a more widespread lineage, but it may need to be recognized with a different name, which surely will require a new gypsum. Here's something else we found along the way, a small digression here. As we were collecting jewelflower fruits, we kept finding this thing. Anybody seen a mustard? It's definitely saliques. Those are mustard fruits. They're long and skinny, but they all point downward. Recognize that? There's some things called Calanthes or Guillenia, and the names I'm going to gloss over here because they are also in flux, data awaiting, but we found it repeatedly, and after 4 weeks in the greenhouse of these seeds, this is definitely a jewel flower from this fruit, and this is this unusually shaped, fast-growing, and in fact flowered within just a couple of weeks in my greenhouse. I had an undergraduate student do these great drawings of what we now know as Guillenia lasiophylla, and I think the name's just changed to Calanthes lasiophylla. It's a spectacular mustard. It's called California mustard. In fact, it's only found within the confines of California, as well as can be occasionally found in Baja.
It was formerly one of the commonest plants in California.
Oh yeah.
That's a white?
This one typically has white flowers. They can be on the cream end, and I'm going to show you a couple more pictures here. Because there's another Guillenia flavescens that has slightly larger flowers, and Guillenia lemonii, depending on whether you're Northern California or Southern California, and some great populations of that at Mount Diablo. But what we got fascinated by is we started looking at its DNA on accident, because we had collected those seeds and started looking at DNA. In fact, we found something really unusual, that not only was this not a jewel flower, but that there were 2 different types. Tim Butler here at the UC Jeps Herbarium, they allowed us to go through their Guillenia collection, which it's in all of these counties, some 30 or so of California counties, and all the little dots here are sampled plants from like 23 of those counties. So we got a pretty good coverage across the state, and we just looked, taking the tissue off of these specimens. Tim got quite good at this, in fact, and he's now able to get amplifiable DNA from specimens from 1930 that have been dead now some 80 years or more. And he extracted the DNA, and he looked at a particular couple of genes, and there were 2 very distinct types. And these types, one of them we call it the coastal type, and it's here and here. And the other one we call it the inland type, although it does get out to the beaches in Baja. And they haven't mated with each other for what we guesstimate about 5 million years. We can use the ticking of a molecular clock that accumulates mutations at a constant rate to suggest that the same time that humans and chimpanzees have been split, for example, these two Guillenias that are absolutely indistinguishable in the field or on herbarium sheets have been separated.
So they're cryptic species.
Exactly. These are very cryptic from a human's perspective.
Right.
We believe that they're entirely selfing as the pollen is already exposed in the bud before the bud opens up. There are a couple of contact points that were really fascinating. One where only 7.3 kilometers separate out two specimens of each individual type. But we found no evidence for hybridization between the two types. And another spot over here.
At identical times?
Yes, according to the herbarium sheets. And on field trip, we found them on either side of the little ridge there.
Is it East and
I think we're actually in Contra Costa at that point. But when grown in the greenhouse, here's some leaves, we just threw them on a flatbed scanner. There are, what seems to me, clearly two distinct types, and they can be differentiated now statistically, not just based on the DNA, because that's not very helpful when you're out in the field, but by the distance between the tip of the largest lobe and the center axis. As these coastal ones typically have deeper lobed leaves, and the interior ones, although they're variable, they never have as deeply lobed leaves. But you actually have to make the measurements in millimeters to get that one correct. But I think we have a new lineage of Guillenia lasiophylla. Probably needs a name, as far as I can tell, nobody's ever distinguished the two. So that's exciting for Tim Butler and his partner. I originally bumped into them looking for this Metcalf Jewelflower. So they are on Serpentine, but they're also off Serpentine. So this one would be fall into that tolerator group. So, like I told you before, Metcalf Canyon, land of many uses. Unfortunately, as we were picking up trash in the Metcalf Canyon, as I felt obliged, we found repeatedly people dumping at this culvert. It's one of the pullouts on Metcalf Canyon. And in fact, there was water running there. And there was water running there when I first got there in the spring, and then it was running there in the middle of the summer. And I thought, well, this is very unusual. I was new to serpentine seeps. And sure enough, come middle of summer, I saw my old friend Aquilegia. It was these red-flowered columbines, and they're growing amongst some thistle. You recognize the thistle there, yeah? And it has this unusually pendant white flowers. And there's also poplar in that seep. And if anyone knows if serpentine seeps if poplar is common to be found in serpentine seeps, that would be a fascinating thing to talk about, because poplar is now very popular, no pun intended, amongst for people studying ecological transitions. So I said, I'm going to be here for a while, and I'm really interested in columbines. Let's start looking at what's going on here in the serpentine seeps.
Before we leave the jewelflower,
Yep.
You didn't talk very much about other aspects of those sites where those species are growing. I mean, you looked at the soil chemistry, but I mean, there's all kinds of other aspects that might be associated with those sites, you know.
Sure.
Did you see anything there that would sort of
It's a
differentiate
continuum. I mean, a lot of you have spent a lot of time at Coyote Ridge too, I'm sure, and it's a very fascinating place, but it's relatively homogeneous. I don't see anything dramatically different between Kirby Canyon and Metcalf Canyon that the UTC land provides a barrier, for example, but I'd be more than eager to hear if anybody knows the difference. There was nothing dramatically different. The types of things that we considered were like herbivores, as these flowers may be susceptible to different herbivores. Those pigments sometimes play roles not only in attracting the pollinators, but in keeping other pathogens off of them. But like I said, we see a large number of herbivores in the wet years, but they weren't preferentially choosing whites over pinks, for example. Yep.
While you're on the California mustard, if anybody wants to see it, there's dozens, if not hundreds of them growing off Style Ranch right now.
Excellent.
They're not very big. A few years ago, I found some that were pretty tall, but it was all over.
That's the Styles Ranch switchbacks to
Right.
Santry Park.
Yeah.
Yep.
And what color is that?
I got photos.
Small white flowers,
Yeah.
but you're most likely to recognize the pendant fruits. They are pointed straight down, and they'll start fruiting right away. Okay, I'd like to show you a little bit about columbines before we run out of time. I want to talk a little bit about the adaptation and speciation in columbines, but really share with you some results of a couple of undergraduates' hard work. Again, Amelia Bethke, a student of mine, did this line drawing, or this colored artwork. She's quite adept at it, and she's done a great job at picking out the distinctive morphological differences between the serpentine columbine and what many of us probably know as the crimson columbine. This columbine grows from Baja all the way to Alaska, and it grows all over western North America. This one here, limited to just a couple of counties in central coastal California. Some of the distinctive differences that I'll mention and we'll see in action later are the cut back openings to the spur. Columbines have these nectar spurs. They're actually the modified petal with a nectar at the base which secretes sugar water. And because of the viscosity of the sugar water that doesn't drip out the bottom, in fact about 20% sucrose is what's in there, and hummingbirds will flutter from below and drink the nectar. There's no landing pad here, so bees are not supposed to come to these, although we do have a lot of videotape footage of them landing on the anther column. This is where the pollen is produced and pollination occurs. Here they have a large blade, we call it, and here they have a very cut back blade. Second of all, there's a very long anther column in the serpentine columbine and a relatively short anther column in the crimson columbine. This is stolen directly from the Jefferson interchange with the Berkeley mapper, just to show you where the serpentine columbine's been collected in comparison to the very widespread Aquilegia formosa. The main point is that there is a dramatic amount of overlap right here, centered in Santa Clara. So I thought, great opportunity to study the process of speciation. In fact, part of my dissertation work showed that the serpentine columbine and the crimson columbine are the closest relatives amongst all the rest of the columbines. Colorado columbine, even deeper, some columbines from Alaska, and then you get the old world columbines, a deeper branch. So these species are very recent, and they're closest relatives. So it's a great window into the process of speciation. Where do new species come from? We might learn something from that dump site on the side of Metcalf. Here's the seep once you get past the trash at Metcalf Road. I know it's kind of hard to see, but the main point here is that it's shady, it's perennially moist, and the columbines do really well amidst these boulders and up into these little waterfalls. It's a really spectacular little place with a really high diverse density of endemics. And the students are out there helping me survey there. This is over at Henry Cal Redwood State Park, and although it looks similar, in fact this is a trail and there's no water there after the rainy season. We also did quite a bit of work out at Rancho Cañada del Oro, where the same Aquilegia formosa grows in these sort of mixed evergreen forests. In fact, in this case, they actually prefer to grow in and amongst the poison oak branches, which Janine quickly discovered, so she's got tights and long sleeve shirt on. So we're going to focus on 6 populations for the next few minutes. In fact, here at Lexington Reservoir, right on the Jones Trail to St. Joseph's Hill, there is a Formosa and a Eximia population within a quarter mile of each other, both right on the main trail there. You'll see the Formosa flowering here in the next couple of weeks. We have Henry Cal Redwoods because I live in the Scotts Valley area, so that's my backyard. And then we have these 2 populations. This is Rancho Cañada del Oro, where we find the Formosa in the poison oak. And here's Styles Ranch, we just mentioned. And there's the Metcalf Canyon spot. So we have 3 Formosas and 3 Eximia populations. They're not necessarily paired, but they're in the general same area. Hummingbirds could definitely fly between these populations. Amongst doing this and finding these serpentine seeps, I just got really excited. I also did my master's work on monkey flowers. Anytime you see columbines and monkey flowers in the same seep, you're in really good company. Monardella, I think, hiding in the background here. Yeah. Silene, hanging from, this is at Lexington. Silene laciniata? Okay, out of serpentine though. And this is a Stachys, I think, growing at the Metcalf Road. Definitely a serpentine habitat, and then some beautiful Achillea. They're really beautiful little gardens, and they'll go off in the middle of July when you're really longing for spring. Go find one of these serpentine seeps and have a look. Like that trail along St. Joseph's Hill, there's a little spot, a grotto there, and it'll keep you busy for several hours. Let's have a little look at the soil. It's a very similar analysis. We're looking at calcium and magnesium. The graph's a little different because it's now on a logarithmic scale. So 1 means that they are present in equal proportions. The Formosa plants grow in non-serpentine soil, and they have a ratio of about 7 to 8. That's their 8 calcium molecules for every 1 magnesium. That's a very easy, benign soil. These eximia soils are extremely challenging for most plants, as only 1 out of every 10 molecules is calcium. We can also look at nickel here, and you'll see that these serpentine seeps have some like 30 to 40 times more nickel in them than they do in non-serpentine habitats. Interestingly, when we did the entire battery of soil, I'm not showing you any of the other 30 chemicals here to save some time, what we did find that was unusual is a lot of salt in these serpentine seeps. It turns out that water comes from deep, deep where the two plates mix, and in doing so they pass through several ancient seabeds. And the hypothesis from the geologists who helped me interpret this was that they're actually passing through layers of crusted sea salt. And in fact, we can use some isotopic ratios to figure out if that's the type of salt we're picking up. Regardless, when you stop, definitely touch the water. You'll see it's very unusual temperature, and it's often very basic, as it kind of has a slippery, smooth texture to it. You can't differentiate much here, but if you get a chance afterwards, come up and look at my soil samples up here. They're not as wet as these petri dishes, but maybe you can make out a little seedling here. As the first question we had was, can these plants grow on each other's soil? Is there any soil preference? Same question as before, but now we have serpentine and non-serpentine, and in fact there is no difference. Lena and Juliana looked at these plates week after week after week, and they germinate fine. The difference happens when you move them to the greenhouse. Now this is the same mom. So you can imagine plants have moms and dads. In fact, when you go collect a fruit and there's seeds inside, that's the mom. You don't know who the dad is most of the time, right? In fact, in these columbines, the dad is probably another plant, because hummingbirds do quite a bit of good pollination, moving pollen around. So we just know that they are half-sibs. They are half-brothers or sisters, right? But regardless, they're pretty genetically similar, we think. And in fact, the same plant, it germinated, was planted here in serpentine soil. But if it's a formosa, maternal family we call it, then it will not grow. Guaranteed. 100% of the time, which is rarely found in these serpentine, non-serpentine studies. Usually it's a statistic that you try to prove. In this case, it was very easy because it wasn't just that one time. Here we take the eximia maternal family and we plant it in serpentine and non-serpentine, and although it's a little smaller, they survive almost 100% of the time when cared for. This didn't just happen once. Each one of these is the pair. These are half-brothers or half-sisters, and you'll see that there's no doubt about it. It was a very easy study to do, as there's a 100% lethality. All of these eximias survive on their own soil, but in fact, they look a little happier when they are on their formosa soil. Again, this concept that, nature is perfectly fit to its environment may need some reconsidering, because we find that, in fact, if you go screwing up with things, right, and messing with things, that actually plants will prefer alternative soils. But here, they're avoiding competition. And these columbines are not good with competition. Any of you who have gardened with columbines know that they are not good competitors. So there's definitely some distinction in the soil type, but it's only in one direction, as the serpentine columbine can grow on both serpentine and non-serpentine. So what's keeping them separated, especially when they grow right next to each other? Something has to be keeping them from hybridizing, which they'll do also in your garden. And I knew something about the flowering time because it's something very distinctive when you're out there looking for them. You look for formosa, these are all the Aquilegia formosas, the crimson columbines in the non-serpentine soil, and you look for it starting now in March and until about May, but boy, come June, they're really hard to find. This is the percent plants in flower, measured at 3 populations, where I took students out every month, and we counted hundreds of plants, and we counted how many of them had a flower stalk and how many didn't. You'll see that, in fact, a lot of plants never flower. That's one of the characteristics of these long-lived columbines. Yet the eximia, growing in these seeps, flowers at a very different time of the year. All 3 populations spike around July and August, such that there is some overlap, but the majority of the flowering occurs with no overlap. It's very difficult to hybridize when you're not reproducing at the same time. We've also done the same study by marking plants and going back to 5 exact same plants and taking photos from the same angle. It just became very challenging to relocate those plants, and we thought this was a better approach. Yeah.
So do you get the same dichotomy in flowering times when you grow them in the greenhouse?
Good question. Thus my demonstration here for today. We don't yet have the flowers, but they should be flowering here in the next couple of weeks, and we'll know whether it's environmentally controlled, because they're very different environments. One of them has water all year round, the other one dries like a bone come June, July. And so in the Santa Clara outside garden where we're doing this experiment, we're letting them get rained on, and we'll eventually water them so that they all get a homogeneous experience, and we'll know that. And this is in slow motion, so stay tuned. You can actually feel the wind of the hummingbird's wings coming.
Wow. Cool.
For something so graceful as a hummingbird, it's especially violent act. Yet the plants are amazingly well adapted. They're pendant, as the hummingbird tends to hover from below.
It's tipping it up.
Yep.
Yeah.
And so they're actually pushing it as far as they can to get the nectar from the very base.
This is Formosa.
This is Aquilegia formosa in non-serpentine habitat. Okay?
What species of hummingbird?
Thank you. Male or female? Difficult to tell. Here is the Aquilegia eximia at Metcalf. Here are the serpentine rocks in the background. The audio has water trickling.
Yes, but not right next to it. It's down about 5 feet.
Exactly. Same species of pollinator, right?
Yeah.
In fact, within flying distance of your average hummingbird. Such that the hummingbird clearly wouldn't differentiate between the two.
So you talk about flying distances, but Anna's hummingbirds, to the best of my knowledge, are very territorial.
They are.
So they wouldn't normally go, they'd have a few hundred yards max staked out, right?
That's not really true. The males are extremely territorial.
Oh, but the females are not.
Females will go to where they can find food and can escape a male.
Oh, okay.
The males do not.
Their territories overlap.
Yeah,
Male and female.
But the females can have a much
Even
larger-
within your 200-meter rule, or whatever rule you want to say, there are spots like, for example, at Lexington, and I'm sure many spots that I have yet to discover, where these two species will grow within a
Oh, I'm
reasonable
sure,
distance.
yeah.
So pay close attention here to where the pollinator is contacting the reproductive parts. In fact, what color is the breast of an Anna's hummingbird?
Greenish to greenish.
Not when it's been visiting Metcalf Jewelflower. Look at that. That is your first yellow-breasted Anna's hummingbird. In fact, in a moment here, there's this monster silver and black camcorder staring right in the face, only a couple of inches from this hummingbird. They are amazingly willing to visit. This hummingbird will come back into play here, and will stop and have a look at you, and you can see where the pollen is transferred.
Did you say this was in Styles? Or
This is Metcalf Canyon. Who's watching who is the question at this moment. What I'd like to point out here is what I think is something new to science, which is exciting for all of us, I hope, which is that taking these stills, the pollen is clearly transferred on the throat of the Anna's hummingbird in Aquilegia formosa in Henry Cowell redwoods. But repeatedly we see the breast of the bird, if not the downright belly of the bird, contacting the reproductive parts and the pollen being transferred much lower down. Such that although the same pollinator could be drinking nectar from these two species,
Oh, they might not pollinate.
pollen would not be transferred, and you would exclude hybridization by the placement of the pollen.
Right.
Now that part, I have to be honest, is not new to science. Some amazing South African scientists have found orchid bees that will have pollinia, the unusual package of pollen in orchids, placed on the head in one species that grows with the same species that puts pollinia on the butt or on the legs. But the placement of pollen in these on hummingbirds in different locations is never previously reported. Here's the still from that where you can really make out the density.
The obvious question is that if the pollen gets attracted to the breast, how is it deposited into the ovary?
Yes, so the stigma, that's the female reproductive part of the pistil, has to have small papillae, those are like hairs, which are more attractive to the pollen. They have small, sticky hooks on them, basically, than the breast of the bird. So there's sort of three tiers. There's the pollen coming off of the stamens, that needs to come off easily, but not too easily or the wind will blow it. Then there needs to be an intermediate stickiness, which is the feathers of the bird, which I've got one right here, and you're welcome to examine, in fact. And then there needs to be an extremely attractive pistil. You see the tears there? And you could actually measure that, but I have yet to.
Where is the pistil in relationship to?
Yeah, it actually emerges from the center of this anther column at a slightly later time period than when the pollen is dehisced. It'll sort of erupt out of the center there, and eventually, if the pollen doesn't get on it, it'll curl back on itself, kind of like the hat of a jester, with usually five, sometimes six. They can self-pollinate, but only after, if they haven't gotten pollen from the top, they will wrap over onto the anthers, which will then sometimes leave whatever pollen remains. And so in your gardens, for example, if you have no hummingbirds, if you have the blue ones and there aren't any bees, you'll still get substantial seed set. But I want to show you something else. Let's see if this is going to work. Oh. This is, again, slow motion. And I said the placement of the pollen was unique. That is, placing the pollen on the breast versus the forehead. But you'll see a hummingbird coming here.
Is the Exenia listed species?
At this point, Anna's hummingbird is transferring pollen on its back. Do you see the gymnastics? This is yet to be reported in any literature that I know. The idea that pollen could be transferred not only on the belly, but also on its back. We have the same flowers now watched for 2 or 3 hours, and I suspect the same exact hummingbird, which has this unique behavior, does this regularly.
Aren't we sure it's Anna's?
I can't be. Maybe you can.
It's highly probable. It's got the markings on the throat, not completely, but it doesn't have anything like an Allen's, and you don't get the Allen's over here. This last year you did have some quiet.
We do get the
Black
button.
This is Styles. Styles Ranch.
That's interesting.
It looks like female.
It's not bright enough.
Because it didn't have the red core
I
gem.
don't think it's anywhere near bright enough for a black chin.
Yep. Okay. So this is Lena and Juliana's work. They are undergraduates in the biology department, and these are Amelia's drawings again. Here they are standing in that little grotto off of that St. Joseph's Trail in Lexington. And in fact, they've been really dedicated to this project, and they are excited to continue it on for another year. So I suspect they will be.
And we find where to look along that.
Park at Lexington. Park at
Yes.
Lexington. Walk up the steep hill through the eucalyptus. And then there'll be a split. One of them takes you to the top of St. Joseph's Hill. Don't go that way. That's too much work. Just continue on. And as you go right past that trailhead, look on your right for Formosa in the next couple weeks. And then wait until June or July and go another half mile and look on the right, and you'll see this grotto. And there'll be water trickling through a culvert in the middle of July, and it'll catch your attention. You'll see that Silene here. You'll see monkey flowers, Mamulus cutatus, the serpentine one, maybe nudatus, depending on what you call it. So, I just want to make sure I thank some of the people who have been instrumental in the Metcalf Jewelflower, particularly the Department of Interior, as they did the majority of the funding for that. And here are the serpentine columbines, in particular my nephew who came up for the pollinator movies. He's a bird fanatic, and I thought it was a great way for us to put together our interests. And Lena and Juliana, who are really passionate about these columbines, they maybe could show you around at some point in time. With that, if you guys have more questions, or be sure to come by and have a look at what I brought in.